field application scientists on Metabolon's international business team. The case study I would like to present to you today is a study which examines the stratum corneum of atopic dermatitis patients. I chose this study because it utilizes the D-squam discs, that is a non-invasive sample collection technology. But I also chose this study because it is where stratum corneum lipidomics was performed, as well as the fact that the researchers in this study were very successful in correlating the metabolomics results to changes in skin microbiome composition. The title of the study that I will be presenting is Stratum Corneum Lipidomics Analysis Reveals Altered Ceramide Profile in Atopic Dermatitis Patients Across Body Sites with Correlated Changes in Skin Microbiome. It was published by Emmert et al. In September 2020 in Experimental Dermatology. Now, what is the background to this study? Atopic dermatitis is driven by the interplay between a dysfunctional epidermal barrier and a skewed cutaneous immune dysregulation. Now, as part of the complex skin barrier dysfunction, abnormalities in lipid organization and microbiome composition have already been described. The objective of this study was to systematically investigate the composition of the stratum corneum lipidome, skin microbiome, and skin physiology parameters at 3 different body sites in patients with atopic dermatitis compared to healthy volunteers. So as I said previously, the researchers in this study set out to systematically analyze the epidermal lipid composition at different body sites in AD as compared to matched healthy individuals. They further took advantage of existing 16S rRNA data to evaluate its potential impact on the skin microbiome composition. So we were able to measure 361 skin lipid species at 3 different sites, namely the forehead, the cubital fossa, and the proximal lower arm, in 4 different groups matched for age, sex, presence or absence of atopic dermatitis, and presence or absence of the FLG haploinsufficiency. In fact, alterations of single or multiple factors impacting barrier function can be mediated through primary mechanisms such as mutations in the gene encoding filaggrin, or indirectly through type 2 immune activity in the skin, causing a secondary downregulation of skin barrier genes and stratum corneum lipids. It has been previously shown that the knockdown of FLG in skin model leads to increased expression of secretory phospholipase A2, which converts phospholipids into fatty acids. In turn, free fatty acids accumulate and contribute to less ordered intracellular lipid lamella and higher permeability in FLG knockdown skin constructs. As I said, we measured 361 skin lipid species, which covered 3 lipid classes, namely free fatty acids, ceramides, and cholesterol sulphate. Ceramide subclasses were further categorized into 10 subclasses according to variations in their fatty acid carbon chain and the sphingoid base architecture. Now, the study revealed that the total abundances of free fatty acids, ceramides, and cholesterol sulfates are increased in atopic dermatitis patients, and that lipid composition differs between body sites. To assess which factors are the major components of variance across all samples, researchers used a hierarchical clustering approach, which is what you see on the left. That is the heat map of ceramides in healthy individuals and patients with atopic dermatitis. The colored bar at the top indicates ceramide subclass composition. The colored bar at the side indicates patients with atopic dermatitis with FLG mutation in red, patients with atopic dermatitis without FLG mutation in orange, healthy individuals without FLG mutation in black, and healthy individuals with FLG mutation in gray. Analysis of variance showed that samples cluster mainly according to atopic dermatitis versus control, rather than FLG mutation versus wild type, confirming that atopic dermatitis is the major component of variance across the data. In fact, notably, samples then cluster according to skin sample site. What you see here is the ceramide and free fatty acid distribution in healthy controls at 3 different sites. Again, highlighting the differences between body sites. This slide highlights how lipid composition between body sites shows significant differences in abundance of ceramide subclasses and abundance of the saturated fatty acid 24O. As the data showed that both arm sample sites are similar, and in order to increase the power of analysis, researchers pooled the data of both the lower arm and the cubital fossa to generate a forearm sample group. Differential analysis of the forehead and the forearm clearly shows that the proportion of the ceramide NS and the ceramide NH are regulated differently. Comparison of healthy individuals and atopic dermatitis patients showed that the ceramide NS group is upregulated in the forehead in both healthy and atopic dermatitis patients. And this is what you see in the graphs to your left. In the graph on the right, you will appreciate the differential abundance of the single ceramides of the ceramide NS and ceramide NH subclass in healthy controls and patients with atopic dermatitis at forehead compared to the arm. The fold change is shown by ceramides with a significant false discovery rate, and only 5 ceramides in both ceramide subclass groups were responsible for the site-specific differences in ceramide class proportions. Now, in this slide, you will be able to appreciate how the researchers in this study were able to correlate the metabolomics results to the 16S RNA data that they were able to obtain, and in particular, how they were able to show that microbial colonization is shifted towards Staphylococcus in patients with atopic dermatitis. In fact, analysis of the skin microbiome composition at the sites of lipid analysis by 16S sequencing showed a decrease in abundance of Bacteroidetes and Actinobacteria, and an increased abundance of Firmicutes in all atopic dermatitis sites. A more detailed analysis revealed a decreased abundance of the genera Chrysobacterium, Propionibacterium, and Cocuria, an increased abundance of the genera Staphylococcus and Lactobacillus, indicating that in atopic dermatitis there is a shift in the bacterial community favoring Staphylococcus and Lactobacillus. A correlation analysis between bacterial and lipid abundances showed a strongly positive correlation of Staphylococcus and ceramide classes AS, ADS, NS, and NDS in patients with AD, but not in healthy subjects. In contrast, the abundances of Propionibacterium, Rhotia, Actinomyces, Lactobacillus, Acinetobacter, Haemophilus, and Neisseria were negatively correlated with the ceramide species AS, ADS, NS, and NDS. In healthy subjects, only Chrysobacteria showed a strong positive correlation with the ceramide species AH, AS, and AP, while other bacterial genera showed only weak positive correlations. These results indicate a correlation of ceramide species with an increased bacterial colonization with Staphylococcus and a decreased colonization with other bacterial genera in atopic dermatitis, but not in healthy subjects. Further, in atopic dermatitis patients, the abundances of Rhotia, Actinomyces, Lactobacillus, Acinetobacter, Haemophilus, and Neisseria showed a strongly positive correlation with abundances of saturated short-chain fatty acids, such as fatty acid 16O and fatty acid 18O. The short-chain fatty acids, free fatty acids, FA16O and FA18O, were also negatively correlated with Staphylococcus in atopic dermatitis patients, thus showing both a negative correlation with Staphylococcus in atopic dermatitis and a positive correlation with bacterial genera other than Staphylococcus in healthy volunteers. Finally, to conclude, the study was able to show that the lipid composition differs significantly between body sites and between atopic dermatitis patients and healthy individuals. Ceramide species NS was significantly higher in atopic dermatitis compared with healthy volunteers, and was also higher in atopic dermatitis patients with an FLG mutation compared to atopic dermatitis patients without the FLG mutation. The correlation analysis of skin lipid alterations with the microbiome showed that the Staphylococcus colonization in atopic dermatitis is positively correlated with the ceramide subspecies AS, ADS, NS, and NDS. This is the first study ever to reveal site-specific lipid alterations and correlations with the skin microbiome in atopic dermatitis.